Okay, so welcome back to this next video in which we are discussing the mammalian circadian timing system. Okay, so we've discussed that in all cells of your body you will have a circadian clockwork going on. Okay, so you have these four proteins, period 1, period 2, cryptochrome 1 and cryptochrome 2, and their levels go up during the daytime and then down during the nighttime, up during the daytime, down during the nighttime, up during the daytime, i.e. they oscillate on a circadian basis and they allow the cell to know what time of day it is. Okay, so we're currently looking at the mechanism by which you actually achieve this oscillation in the level of period proteins and cryptochrome proteins. Okay, right, and it's what is known as a transcriptional translational feedback loop. So we're going to start off at this point where you have very low levels of both period proteins and cryptochrome proteins, and we're going to see how you get this rising phase. How do you produce more period and cryptochrome proteins? Okay, so you're going to have transcription factors which are going to promote the expression of the period genes and the cryptochrome genes. So what are these transcription factors? Well, basically they are heterodimers of two separate proteins. Okay, so let's discuss the proteins that are going to be involved. So the first protein that is extremely important in maintaining this circadian clockwork is a protein called BMAL1. And what's going to happen is BMAL1 is going to associate with another protein, okay? And the two of them are then going to act as a transcription factor that's going to promote the expression of the period genes and the cryptochrome genes. Okay, right. Now, there are two different binding partners that you can have here to bind to BMAL1. You can either have a protein called NPAS2 here, okay, or alternatively you can have a protein called clock. Both of these proteins can dimerize with BMAL1, and this heterodimer of either BMAL1 with NPAS or BMAL1 with clock is then capable of activating the expression of period genes and cryptochrome genes. Okay, so the BMAL1 NPAS2 heterodimer or the BMAL1 clock heterodimer is going to now act as a transcription factor. So let me remind you of what a transcription factor is. Okay, so let this let these two lines here represent a piece of double-stranded DNA. Okay, and let's say we have a gene here. Okay, so this section of the double-stranded DNA here, let's say this is a gene for either a period protein or a cryptochrome protein. So this portion that I'm boxing in turquoise here, this is either a gene for period, a period protein, and I'll put capital PER for period, or it's a gene for a cryptochrome protein, and I'll put capital CRY, cry, for a cryptochrome protein. Okay, so it's one of the two. Okay, now, upstream of all genes in eukaryotic cells, not just these period genes and these cryptochrome genes, upstream of all genes you have a special region known as the promoter region. Okay, so I'll highlight this portion in purple here. Okay, and this portion is not involved in being translated, okay? So it's not going to be turned into protein. However, it is involved in controlling the expression of the downstream gene. Okay, and what do I mean by that? Well, I mean how much of the gene product of this gene you're actually going to produce. Now, why is the promoter region so important in controlling that? Well, it's because the promoter region is where the RNA polymerase II enzyme binds to the DNA and then works its way along the DNA to synthesize a piece of mRNA. Okay, so the promoter region is where the RNA polymerase II enzyme, which I'll abbreviate to RNA for RNA, P for polymerase, and then 2 for 2. This promoter region is where the RNA polymerase II will come, bind, and then open the DNA, work its way along the coding strand of the DNA, and then produce a piece of mRNA that's complementary to that. Now, if the promoter region has a very high affinity for RNA polymerase II, i.e. RNA polymerase II loves to bind to this promoter region, then RNA polymerase II will bind there all the time. You will then get a huge production of mRNA for this gene, and then that mRNA will translate into a large amount of the protein. Okay, so you'll get a lot of protein being produced. On the other hand, if the promoter region has a very low affinity for binding RNA polymerase II, then RNA polymerase II will hardly ever bind here, okay? Which means you'll get hardly any mRNA being produced for these downstream genes. And that means that you'll get hardly any protein being produced. So the promoter region, by determining its affinity for RNA polymerase II, controls how much mRNA of the downstream gene is actually produced, and therefore how much protein is actually produced. Okay, right, so that's what a promoter region is. Now let's talk about what transcription factors are, and we'll do it firstly in full generality, and then we'll come to these BMAL MPAS2 or BMAL1 clock heterodimers. Okay, so basically a transcription factor, which I'll show here, and we'll have it in orange. So in orange here, this is meant to represent a transcription factor. A transcription factor is a protein which combines to promoter regions, okay? And it won't just bind to one promoter region, it'll bind to the promoter regions upstream of a huge plethora of genes, okay? So usually transcription factors will bind to the promoter regions of hundreds of genes, okay? So it will bind to the promoter region of many, many genes, and at these promoter regions, what it will do is it will change the affinity of that promoter region for binding RNA polymerase II. And at some of the promoter regions that the transcription factor binds to, it will increase the affinity of the promoter region for RNA polymerase II. Whereas at other promoter regions, for other genes, remember I said the transcription factor binds to the promoter regions of a whole plethora of genes, okay? At some it will bind to their promoter regions and increase their affinity for RNA polymerase II. At others, it will bind to them and decrease their affinity for RNA polymerase II. Now, what's the significance of this? Well, if a transcription factor binds to a promoter region and increases the affinity of that promoter region for binding RNA polymerase II, then RNA polymerase II will bind them more often. It will therefore produce more mRNA, okay? And when you've got more mRNA, you'll then make more protein. So, if a transcription factor binds to a promoter region and increases its affinity for RNA polymerase II, it has hence enhanced the expression of the downstream gene. Okay, so it was a transcription factor enhancer. Okay, if on the other hand a transcription factor binds to a promoter region and decreases the affinity of that promoter region for RNA polymerase II, then you will get RNA polymerase II binding there less often, you'll get less mRNA being produced for the downstream gene, and therefore less protein. Okay, therefore it will have repressed the expression of the gene. Okay, so transcription factors can enhance the expression of some genes and then repress the expression of other genes. So for any transcription factor, it has this whole plethora of promoter regions that it binds to. At some, it will be a transcriptional enhancer. Okay, so it will increase the affinity of that promoter region for RNA polymerase II. At others, it will be a transcriptional repressor, and it will decrease the affinity of that promoter region for RNA polymerase II. Okay, right. So, we then are discussing these heterodimers of BMAL1, which I think I'll colour in green here. Okay, and then it's either dimerized with MPAS2 or clock. Okay, shown here in blue. Right, so what will happen is these BMAL1 MPAS2 or BMAL1 clock heterodimers will act as transcription factors. Okay, so they'll bind to the promoter regions of a whole plethora of genes and change the expression of those genes. Now, some of the genes that they bind to the promoter region of include the period and cryptochrome genes. Okay, so what will happen is our BMAL1 MPAS2 or BMAL1 clock heterodimer will bind to the promoter region upstream of period period genes and also cryptochrome genes, and will increase their expression. Okay, so they're going to act as enhancers by increasing the affinity of the promoter region of those period or cryptochrome genes for binding to RNA polymerase II. Therefore, you'll get RNA polymerase II binding more often, and you'll get an increase in the production of those proteins. Okay, so this is going to lead to you producing more and more period proteins. Okay, and period, remember, includes period 1 and period 2. Okay, so I'll just put period, but remember that means both period 1 and period 2, but as far as we're concerned, they do exactly the same thing. And then also more cryptochrome proteins, and once again I will just put cryptochrome rather than stressing the different forms of it, because again, as far as we're concerned, they do exactly the same thing. Okay, so let's colour these two proteins in. So we'll have period in red here, and then we'll have cryptochrome in yellow. Okay, right. So what then happens is period and cryptochrome are going to go up. Okay, so you're going to get rising levels of period and cryptochrome. So we're now in this phase of our graph. We're going up, up, up. Now, what happens is as these proteins get higher and higher within the cytoplasm of the cell, they start to aggregate into complexes. Okay, and this isn't fully understood, but what we do know is that they seem to form complexes which have a totally different function from the individual proteins. Okay, so they seem to form some sort of large complex. So I'll just sort of show a complex like so. So here is our complex, and this complex will go back into the nucleus. Okay, so it's going to go from the cytoplasm into the nucleus, and it's going to stop the BMAL1 MPAS2 or BMAL1 clock heterodimers from being able to activate the expression of the period or cry genes. Okay, so it goes into the nucleus and it inactivates these BMAL1 MPAS2 or BMAL1 clock heterodimers. It does something to them, binds to them most likely, and just stops them from then being able to activate the promoter regions of the period and cryptochrome genes. Okay, so your production of period and cryptochrome is then going to go down. Okay, and it's all because of the formation of these large complexes here. Okay, which I'll just show in orange like so. Right, okay, so let's discuss why this then gives rise to this sort of a oscillating level of period and cryptochrome. Well, as the period and cryptochrome go up and up and up, you're going to get the formation of these complexes in the cytoplasm, which then are going to go back into the nucleus, and then they will stop these BMAL1 MPAS2 or BMAL1 clock heterodimers from being able to activate the expression of period and cryptochrome. So you're going to stop producing any more period and cryptochrome. Okay, so we've cut off production. Now what will happen is gradually the period and cryptochrome proteins will be broken down. Okay, so then levels within the cell will start to fall. Okay, and that's this down phase of this oscillation here. Now, as the levels fall, that will lead to the complexes also going down. Okay, so the complexes will deassemble gradually, and they will go down within the cytoplasm, which means that they will stop blocking the BMAL1 MPAS2 BMAL1 clock heterodimers from being able to activate the expression again, and now we're back to the beginning. Okay, we're back to square one. So now BMAL1 MPAS2 or BMAL1 clock heterodimers will be able to activate the expression of period and cryptochrome, and will begin another cycle. And overall, this cycle takes 24 hours. Okay, that's the magic number for this cycle. Right, so this is known as a transcriptional translational feedback loop, because we are getting the transcription and translation of the period and cryptochrome proteins, Okay, that's the transcriptional translational portion, and then the production of these proteins are then feeding back on the transcription and translation, specifically the transcription, and they are stopping the production of any more. So by producing more protein, the protein then stops its own production basically, and that then leads to the levels of the protein going down, which then means it no longer inhibits its own production, which means that you produce it again, and then you get this oscillating level. Okay, right, and overall the magic of this pathway is that it takes 24 hours to go around in a cycle. Okay, so that's the main transcriptional translational feedback loop that is responsible for this circadian oscillation in the levels of period and cryptochrome within the cytoplasm of a cell. Okay, right. So what we now want to look at is an accessory transcriptional translational feedback loop, okay, that also helps this, because at the moment you might be thinking that, okay, the BMAL1 levels will be constant within the cell, never mind N-PAS2 and clock, okay. You might be thinking that BMAL1 levels will remain constant throughout this entire process. Yes, okay, the complexes of period and cryptochrome will come in and stop BMAL1 with its binding partner from activating the increased expression of period and cryptochrome, but they're not destroying BMAL1. So you might think that BMAL1 itself is not going to oscillate, however, you'd be wrong. And what we now need to discuss is an accessory transcriptional translational feedback loop that also helps to keep period and cryptochrome levels oscillating within the cytoplasm of the cell. Now, you can knock this accessory loop off and period and cryptochrome continue to fluctuate. So the pathway that we have looked at is enough to get oscillating levels of period and cryptochrome. However, there is an accessory pathway that just helps it out a little bit. Okay, right. So, basically, we'll start again at 0 hours. Okay, so we have our BMAL1 protein here. Okay, with its binding partner, which is either N-PAS2 or clock. Okay, so here is BMAL1, and it's bound to either N-PAS2, and I'll try and fit these in, N-PAS2, or it is the clock protein. So those are separate proteins, but they both perform the same role, basically. They are the binding partner for BMAL1. Let's add a little bit of colour on here. So N-PAS2 and clock can be represented in green here, and we'll have BMAL1 in orange in this picture. Right, so we know that these heterodimers here are responsible for binding to the promoter regions of the period and cryptochrome genes, but they also bind to the promoter region of another gene, okay? So let's have a piece of double-stranded DNA here, and I can use this as an opportunity to tell you a little bit more in-detailed information that I forgot in the previous discussion. Okay, right, so we'll have our gene here, and this gene is going to be for a specific protein known as Rev alpha. Okay, so this is Rev and then alpha gene. Again, it's going to produce the Rev alpha protein. Now, of course, the Rev alpha gene has a promoter region, like all genes within eukaryotic cells. And what's going to happen is BMAL1 N-PAS2 / clock heterodimers are going to bind to this promoter region. Now, the bit of information that I forgot to tell you when we were discussing it previously was the exact sequence of 6 organic bases that you have to have present within your promoter region in order for BMAL1 N-PAS2 / clock heterodimers to bind to you. Okay, so if you want these heterodimers to bind to your promoter region, then your promoter region has to have a certain sequence of 6 organic bases. Okay, so within your promoter region, shown here as this little section here that I'm now highlighting in blue, you have to have a sequence of 6 organic bases known as an Ebox. Okay, so what is this sequence then of 6 organic bases? Well, basically the sequence that you have to have, if we zoom in on this blue portion here, okay, so let's say this is our little blue portion zoomed in on. Okay, now I'll draw the complement. Oh no, I haven't left enough space for that, damn. Let's draw another picture, because I can't, well actually I can show it on one strand. I won't be able to show the complementary strand though, never mind. Okay, I won't be able to fit it in. So here is the first organic base in this Ebox is a C. Okay, next you have another an A. Then you have C. Okay, then you have G, and then a T, and then a G there. Okay, so that's the sequence that you need to have within your promoter region in order that the BMAL1 MPAS2/clock heterodimer can bind to you. And this sequence of 6 organic bases, like so, is known as an Ebox. Okay, right, so this is the Ebox. All promoter regions that bind this transcription factor will have one of these Eboxes. So the promoter regions in all of the period and cryptochrome genes will also have these Eboxes. I just forgot to tell you at the time. Okay, right. So the promoter region of the Rev-ERB alpha gene also has one of these Eboxes, and therefore BMAL1 MPAS2/clock heterodimers combined to the Ebox within this promoter region and then increase the expression of the downstream gene. Okay, so you're now going to produce a lot of Rev-ERB alpha. Okay, so whilst we're producing period and cryptochrome, okay, so in the up phase of this cycle, so like so, this is our 24-hour cycle, so here's 24 hours, here's 0. This is the level of period and cryptochrome, which I'll just abbreviate to per and cry. Okay, so whilst this is going up, Rev-ERB alpha is also going up. Okay, now what does Rev-ERB alpha do? Well, Rev-ERB alpha actually causes the levels of BMAL1 within the nucleus to go down. Now, how does it cause this? Well, it's going to prevent the transcription of BMAL1. So, if we draw the gene for BMAL1, okay, here, like so, so this is the BMAL1 gene here, then it's going to prevent the transcription of BMAL1, basically, so that you lower the levels of BMAL1 within the cytoplasm, well, within the whole cell, basically. So BMAL1 levels are going to now go down within the cell. Okay, right. Now this helps with the entire period cryptochrome cycle because, remember, as period and cryptochrome go up, Rev-ERB alpha will also be going up. And that means that Rev-ERB alpha going up is going to mean that BMAL1 goes down. So if this is the graph for period and cryptochrome here, and we'll have this in turquoise, okay, like so, then the graph for BMAL1 is going to be doing this at the same time, basically. It's going to be going down because as period and cryptochrome go up, Rev-ERB alpha is also going up because they're both activated by the same thing. Okay, so BMAL1 goes up. So not only do period and cryptochrome stop their own production by stopping BMAL1 MPAS2/clock heterodimers from activating their transcription, but Rev-ERB alpha is actually leading to BMAL1 going down anyway. So both of these things are going to help lead to the production of period and cryptochrome reducing, basically. Okay, now, of course, when BMAL1 then goes down, what will happen is you will no longer be getting the activation of the production of Rev-ERB alpha. So then Rev-ERB alpha will also fall because you stop producing it, but you continue destroying it. Okay, so Rev-ERB alpha will be doing the same thing as period and cryptochrome here, so I'll add it on here. Okay, so it will start to fall because the amount of BMAL1 has fallen and therefore its production has gone down. When Rev Herb alpha starts to fall, of course, that means that BMAL1 transcription will start up again. So BMAL1 will start to go up again, and this takes us back ready for the next cycle. Then when BMAL1 goes up, you'll start producing more Rev Herb alpha, and this will go down again. And this potentiates the period cryptochrome cycle because BMAL1 is now going down when period and cryptochrome production needs to stop, basically, i.e. when period and cryptochrome have reached a peak and you now want it to go down. Okay, so this is going to potentiate the period cryptochrome cycle. And again, this Rev Herb alpha and BMAL1 levels, these will cycle on a 24-hour basis, but you can see that the levels of BMAL1 are cycling completely antiphasically to the levels of period and cryptochrome, but it's not directly. Period and cryptochrome do not cause the levels of BMAL1 to go down. It's Rev Herb alpha, which is also oscillating in the same way as period and cryptochrome, which then causes the levels of BMAL1 to go down. Okay, right. So that then is the circadian clock within human cells. Okay, in the next video what we'll start with is a discussion of what the purpose of having a circadian oscillator like so is, and then we'll just start to discuss how we synchronize all the cells of the body so that they have the oscillator all in phase, basically.